Hello everyone, welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on progressing an antibacterial drug discovery project and an SME perspective. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All of our webinars are recorded and freely available to view after the live session on our website, revive.gardp.org/webinars. You can watch the webinar we previously held on the View from Industry and Academia on progressing antibacterial drug discovery projects here as well. You can also read our series of articles known as Antimicrobial Viewpoints, where experts in the field discuss various topics. We also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions on various important terms, and some of these also include videos where experts give further explanations on the terms. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window, as shown on the slide. We will address the questions after the presentations and do our best to respond to as many questions as possible. If your question is for a specific speaker, please do indicate your name at the beginning of your question. Today's speakers are Alisa Serio and Victoria Savage, and our moderator is Daniel Ritz. Daniel Ritz is Senior Director and Senior Group Leader in the Drug Discovery Translational Science Division at Idorsia Pharmaceuticals Limited. He has over 20 years of experience in industry and previously led the preclinical anti-infective group at Actinium, where he worked on cadazomid, which was in clinical development for the treatment of C. difficile infections. Daniel received his PhD in microbiology from ETH Zurich in Switzerland and completed a postdoctoral fellowship in the lab of John Beckwith at Harvard Medical School in Boston. He has co-authored more than 30 papers on the topic and holds several patents. Welcome, Daniel, and I'll hand over to you to introduce our first speaker.
Thank you. Our first speaker today is Alisa Serio. Alisa is Executive Director of Microbiology and Non-Clinical Development at Paratek Pharmaceuticals, a mid-sized enterprise located in Boston, Massachusetts. She has over a decade of experience in antibacterial research and development, in particular on combating antibacterial resistance, and has contributed to the FDA approval and commercial launch of new antibiotics, as well as research activities under the US government partnerships. Alisa has received her PhD in molecular biology and microbiology from Tufts University Graduate School of Biomedical Sciences, and has completed a postdoctoral fellowship in molecular and cell biology at the University of California in Berkeley. And I'm happy to welcome Alisa. The floor is yours.
Thank you very much. All right, so I'm happy to be here today to tell you about progressing an antibacterial discovery project from the small, medium, and enterprise perspective. Okay. So the views and opinions in this program are my own and don't reflect the views and opinions of my employer. I also want to point out that everything I'll tell you about today is non-confidential and non-proprietary. There are a number of references that I provide at the bottom that provide good resources for additional information if you're curious. Okay. So first I'll give an overview of the drug discovery and development process through approval and commercialization and highlight just a few key considerations. And then I'll go right into examples of go, no-go decisions and successful approvals of programs that I've worked on, with a particular consideration for program origination, program strategy, and how and when funding can play a role. And this is all within the SME perspective. My experience is solely on small molecule programs, and so that'll be the focus today. I think that the key takeaways, though, can apply to any modality. This is a big picture of antibacterial drug discovery and development. through all of the phases, included not only early drug discovery, when we think about identifying novel targets or confirming known targets, compound screening, selection of those lead compounds through an optimization process, preclinical gating studies that are in vitro and in vivo designed to make those go, no-go decisions, and then ultimately selection of a candidate, putting that through IND enabling studies and studying now your candidate in clinical trials, ultimately submitting to a regulatory authority for gaining regulatory approval. And then there are a number of post-approval activities that I think are important to consider as well. Not only that commercialization step where you want to get your drug to the patients in need, but there are also post-marketing requirements typically following approval by your regulatory agency, phase 4 studies, potentially other regulatory submissions, and exploration of other indications. And so the companion webinar recently provided by Ken Bradley and Dermot Hughes gave the large pharma, large industry, and academic perspectives on the same topic. And a lot of those key learnings will apply here. So I won't go into the whole list of the types of gating studies for go, no-go decisions and preclinical. That'll still apply in this case. They also talked about target candidate and target product profiles. And of course, that's very important at all stages of your discovery and development program. So I think really understanding what you're seeking, who the patients that you're seeking to treat at the beginning, and continuing to check that throughout your sometimes decades long development program is critical because perhaps the patients that you set out to treat, are they relevant at the state of approval, at the time in which you're going to commercialize your drug? So important things to consider. The examples I'll talk about today are going to be at all of these various stages. So let's go into that. So I'll be discussing 4 programs that I've worked on. The first 3 programs listed are programs I worked on at Achaogen, and the last is omadacycline, a Paratek product. And again, I'll be focusing on the foundation of these programs, how that led to the strategy, and how that then led to funding opportunities. The first program I'll discuss is the biotin carboxylase inhibitor program, and this slide captures its foundation. The foundational work was conducted at Pfizer, and the Miller et al. Reference at the bottom is a wonderful example of a discovery program that was able to discover a novel target, novel molecules, and then conduct proof of concept studies. So here the authors reason that eukaryotic compound libraries may be a potentially untapped resource for antibacterial compounds. Now, traditionally, we want to stay away from any compounds that might have cross-reactivity with eukaryotic targeted compounds. We don't want to have any potential issues, but they rationalize that it's possible that there were compounds in this 1.6 million compound library that actually could be selective for bacterial targets over eukaryotic targets. And so the authors of this work performed a high throughput whole cell screen using an E. coli strain that was membrane deficient and efflux deficient, and as a result, identified compound 1, which is a pyridopyrimidine that binds to the ATP binding site of biotin carboxylase, which is a member of the fatty acid biosynthesis pathway in bacteria. So here they were able to identify a new antibacterial target and compound class, and then put that compound through a series of proof of concept studies, which are listed on the right side. And here we see that they confirmed in vitro activity against gram-negative pathogens, including not only E. coli, but also Pseudomonas aeruginosa and Haemophilus influenza. They were able to demonstrate that the compound was able to co-crystallize and interact with biotin carboxylase by a crystal structure, defined target resistance mutations, confirmed that this compound targeted the fatty acid biosynthesis pathway via macromolecular synthesis assays, demonstrated cyclicality, and studied the compound also in vivo in efficacy and PK studies, conducted plasma protein binding assessment, and importantly, we're able to demonstrate selectivity for bacterial biotin carboxylase versus eukaryotic protein kinases. So that accomplished their goal. Now, this laid a foundation for the biotin carboxylase inhibitor program at Achaogen. So that program then was focused on a medicinal chemistry strategy to perform rational optimization using a structure-guided approach. And subsequently, the optimization was focused on optimizing the physiochemical properties, really to get into the gram-negative bug, so avoid that any membrane penetration issues, and also efflux. And as a result, 19 new molecules were discovered at Achaogen, and that's published in the Andrews et al. Reference, with improved antibacterial potency against gram-negative pathogens. And this foundation and strategy also led to successful funding opportunity. So the work was funded by the NIH. And so I may have inadvertently touched my thing, but we'll go to the next slide. And so following some early gating studies, there was ultimately a no-go decision, and here it's due to a resistance liability. And so while the lead molecules in that series did have improved potency, they, as well as an improvement on the spontaneous mutation frequency compared to compound 1, they did, mutants that were selected did result in MIC shifts, or minimum inhibitory that were alarmingly high. And what do I mean by that? So if we take a look at the table, you can see compound 1, the wild type background for compound 1 was 16 with an MIC against the Pseudomonas strain, and 14A had an MIC of 1, so demonstrating improved potency. But mutants were selected that had MIC shifts up to 256, for example, which is the next line down. And mutations were confirmed in the biotin carboxylase target, or due to efflux overexpression. And so at this point, the program was terminated, and resources were focused to more promising programs. The next program I'll discuss is the LPXC inhibitor program, and this captures at a high level the foundation of the LPXC inhibitor. So LPXC catalyzes the first committed step of LPS biosynthesis. It's essential in most gram-negative pathogens, and there are no mammalian homologs, so it makes it an appealing target. Early work was done at Merck. In a chemical library screen, LPXC inhibitors were identified, and then subsequently British Biotech. It was not until a bit later that it was confirmed that the target was LPXC, and there have been several discovery programs that have investigated LPXC inhibitors, Chiron and Pfizer also included, and this is not an all-inclusive list. So these inhibitors demonstrated antibacterial activity against gram-negative pathogens, including Pseudomonas aeruginosa. So again, this foundation laid the foundation of the LPXC inhibitor program that contributed to the strategy, which was a medicinal chemistry strategy and a research campaign to identify novel LPXC inhibitors that were particularly targeting important gram-negative pathogens, including Pseudomonas aeruginosa. And so a robust preclinical program was completed, and a lead molecule, ACHN975, was identified with potent activity against Pseudomonas aeruginosa. You can see an MIC90 here of 0.25 microgram per mil. Now, again, that foundation and strategy led to a number of successful funding opportunities and awards, including from the Defense Threat Reduction Agency, NIAID, Wellcome, and CARB-X, all of which funded the LPXC inhibitor program at Achaogen at various stages. So ultimately, following a completion of all of those preclinical gating go, no-go studies, an IND was submitted, and ACHN975 was taken into the clinic. Now, as a result of those studies, we did understand a little bit about a potential liability, and that's specifically with resistance. So we'll talk about resistance again here. We understood that a mutation prevention concentration, so that's a concentration that we needed to reach a concentration at least 8 times above the minimum inhibitory concentration of the Pseudomonas strain we were studying to reduce the likelihood of resistance, and higher is always better. And so that led us to this point where we needed to ask, could we achieve a dose regimen that would reduce the potential for resistance but minimize any tox signals that we might see? And that's your typical therapeutic window. Unfortunately, although this was the phase 1 study, unfortunately did need to be terminated and resulted in a no-go decision with ACHM 975 due to tox observed in the study. Inflammation at the injection site was observed, as well as the Cmax-driven dose-limiting toxicity of transient hypotension without tachycardia. So, while this was exciting, it was the first LPXC inhibitor dosed in humans, we unfortunately observed sort of a new tox signal, if you will. And that did not result in terminating the entire LPXC inhibitor program. Instead, the team went back to refocus its efforts on an additional medicinal chemistry campaign with the objective to identify a clinical candidate with a wider therapeutic window for cardiovascular safety. And alternative leads were identified, and this is discussed in depth in the Krause et al., Cohen et al. References. So again, I would direct you there if you'd like to hear more about all the troubleshooting that went on to identify alternative leads. LPXC 516 is shown here. This was the most promising lead, and the leads were put through a focus battery of in vitro and in vivo studies to really hone in on whether or not we had a new lead that could go into IND-enabling studies and eventually back in the clinic. And this was specifically around enzyme potency, antibacterial activity, resistance, PK, and also a therapeutic, could a therapeutic window in a rat cardiovascular safety assay be obtained? Unfortunately, the program did have to be terminated at this stage, so there was a no-go decision because we could not identify a new optimized clinical candidate that had an acceptable therapeutic window. It was just not possible to dose these molecules high enough to prevent that resistance liability that we were observing in vitro. So in both of the examples I just provided, we are thinking about novel targets, novel molecules, and the foundation laid by large pharma predominantly. In the next 2 examples I'll give, they share similarities in that the foundation is based on an existing class. So this is the plasmamycin program foundation and strategy. So here we have an existing class with an extensive history, and that is the aminoglycosides. So aminoglycosides have been used clinically for over 70 years. They are bactericidal protein synthesis inhibitors that are broad spectrum. They have predictable PK, are thought of as soluble and stable, lacking metabolism, and importantly, toxicities are well known and well understood to a degree, and resistance mechanisms are also understood. And so based on this foundation, a strategic medicinal chemistry campaign was built with the purpose of optimizing an aminoglycoside. So here, plasmamycin was modified from the cisomycin scaffold, shown in black, with 2 modifications shown in green. These modifications block aminoglycoside modifying enzymes, which are the most common clinically relevant aminoglycoside resistance mechanisms. So here we now have a new molecule that's able to evade known resistance mechanisms and expand the spectrum. And of course, understanding the class itself can help guide those gating studies and make them more focused. So ultimately, the plasmamycin program completed its development program, and so this is highlighting many years of work in one slide. There are just a few things I'd like to highlight here. So first, The plasmamycin program at Achaogen was awarded the first contract from BARDA, Biomedical Advanced Research and Development Authority's antibacterials program. And this award funded manufacturing, development, regulatory interactions, as well as phase 3 studies. And so here's an example where we have an award later in a discovery program. Plasmamycin was also granted FDA Breakthrough Therapy designation and Fast Track designation. Now, while Achaogen, of course, went through the typical venture capitalist series, it also went public in 2014 and was listed on the NASDAQ. So the phase 3 studies were focused on the indications of complicated urinary tract infection, acute pyelonephritis, and bloodstream infection due to carbapenem-resistant Enterobacteriaceae. Ultimately, the FDA approved plasmamycin only for the cUTI indication in 2018. Subsequently, plasmamycin is marketed under the brand name Zemdry. Unfortunately, Achaogen did go bankrupt in 2019, shortly after this approval, and the plasmamycin asset was sold to Cipla Therapeutics, who just recently gained approval of plasmamycin in India in 2024. Now, the omadacycline program also has a similar foundation and strategy in that here we're thinking about the tetracycline class. So again, tetracyclines have over 70 years of clinical use. So they're an existing class with a long history of safety and utility. Tetracyclines are generally bacteriostatic protein synthesis inhibitors, again with predictable PK. They're thought of as soluble, orally bioavailable in many cases, and have known toxicities and also known resistance mechanisms. So again, a strategic medicinal chemistry campaign can be built to optimize on the knowledge of what we know about tetracyclines and the potential gaps. So the omadacycline structure is shown here. It's an aminomethylcycline with modifications at the C7 and C9 positions that allow it to evade active efflux pumps and ribosomal protection proteins, which are the most common clinically relevant tetracycline resistance mechanisms, while also maintaining oral bioavailability. So omadacycline that went through as well a full development program, and it's captured, again, decades, years of work is captured here in one slide. So I will point out a few things here as well. So one is that throughout the development program of omadacycline, Paratek conducted or entered into successful collaborations and in licensing agreements with a number of large pharma companies. So Bayer, Merck, Novartis, Transcept Pharma, all played a role in helping to move omadacycline through the development program. In addition, Paratek also conducted the series venture capitalist investments and then eventually went public in 2012 and was listed on the NASDAQ. Omadacycline was granted FDA Fast Track designation, and omadacycline was studied in 2 indications in phase 3, acute bacterial skin and skin structure infection and community-acquired bacterial pneumonia, and was approved by the FDA for both indications in 2018 and is marketed now under the brand name Nuzyra. Paratek also entered into a collaborative licensing agreement with Zylab based in China, who subsequently gained approval of omadacycline in 2021 in China. So there's also post-approval activities I'd like to highlight because as you recall in one of the early slides, I showed early discovery all the way through post-approval. And I think keeping this in mind throughout your program is really important. So of course, there are post-marketing studies that will be required typically by your regulatory authority. And then it's also important to keep in mind the potential to develop your drug perhaps for new indications that you may not have studied in your phase 3 trial. And so omadacycline is under development for new indications and was awarded a contract by BARDA through Project BioShield for the development of omadacycline for the treatment and post-exposure prophylaxis of pulmonary anthrax. And so as part of that award, BARDA also supporting the funding of some of the post-marketing required studies. So this is a great example of where you can again have an opportunity for funding even late and post-approval. In addition, and separate from BARDA, omadacycline is also being developed for non-tuberculous mycobacteria pulmonary disease and is granted FDA orphan designation and the analogous designation in Europe. Late last year, Paratek actually went private, was taken private by an acquisition by Point Capital and Novo Holdings. And so that's also important to mention here. All right. So in conclusion, I've highlighted the foundation of 4 different programs at SMEs, and with sort of 2 angles here. So one was a foundation where we have a novel target, novel molecules, less information known, but proof of concept clearly demonstrated. And in those cases, that was primarily demonstrated by large pharma, who, for example, have access to large chemical libraries and can do a lot of that initial work. Those resources are available at that time. Alternatively, another foundation is understanding a well-known class that's been used in the clinic for decades and understanding those liabilities or areas for improvement. And so in both cases, we have this foundation that can lead to a specific strategy around optimization. And that foundation and strategy then can lead to funding opportunities that can occur at all stages and in all various forms. So I've given you examples of funding from non-dilutive funders, grants such as NIAID and partnerships with BARDA, for example, as well as collaborative agreements with large pharma. And so with that, I would like to thank you for attending. I'm looking forward to answering your questions later. I also want to thank all of the authors and researchers that have contributed to the work that I've just presented, as well as all of the funding partners that helped move these programs forward at the SME level. Thank you.
Thank you, Alisa. We're just moving to our next speaker, which is Victoria Savage. Victoria is the Chief Scientific Officer at INFEX Therapeutics, an infection-focused small biotechnology company developing several new therapies for drug-resistant infections based in Alderley Park in the UK. She's a microbiologist with an interest in antimicrobial research and development and has contributed to the development of multiple antimicrobial programs from early stage through to clinical stage projects. Victoria has more than 15 years of experience in the development of diverse modalities, including small molecules, biologics, and non-traditional approaches. She gained her PhD in microbiology and immunology from the University of Leeds in the UK, and also serves on several scientific advisory committees in biological sciences. Welcome, Vicky. Please start your presentation.
I hopefully have control now. Fantastic. Thank you, Daniel, for that introduction. It's a real pleasure to have the opportunity today to give you my perspective really on what it's like working in an SME developing new antibacterial products and really some of the challenges that SMEs can face working in this area. So really, I guess to set the scene, it's worth talking about the broken market in AMR. So developing new antibiotics, much like any other therapeutic area or medicine that you're developing, is an extremely expensive and an extremely high risk endeavor. So it takes around 10 to 15 years at the very least to develop a new antibiotic, and it costs in the region of $1 to $2 billion. And of course, drug discovery is high risk, so a lot of products fall out of development before they get anywhere near a patient. But for antibiotics, even those products that do get through to approval for use in patients, the reimbursement and revenue that antibiotics can make is extremely low. And that is for a couple of reasons. One is that traditionally antibiotics are priced quite low, but also because, of course, if you bring a new and novel antibiotic to market, it is going to be kept in reserve absolutely for those patients who need it, whose infections were not treatable by generic or older antibiotics. So that, of course, is the right thing to do, but of course that means a low volume of sales for new antibiotics entering the market. So on the image on the left-hand side, so this is just taken from an article from the Pew Trust website, and we can see that the combined sales of all branded antibiotics in the US in 2018 was $535 million. And of course, that may sound an awful lot of money, but when we consider this against other therapeutic areas, so if we take the arthritis drug Humira, for example, this made sales of greater than $19 billion in the same year. And this low revenue, unfortunately, has led to the demise of numerous developers in this area. So again, in the image on the left-hand side, we can see that 5 of the 15 developers of new antibiotics that were approved since 2010 have since collapsed. And the image on the right-hand side is just taken from the Wellcome Trust website. So this is a nice article on the broken market in AMR. So the links for these are just at the bottom, should you wish to go away and have a look at these articles. But the image on the right really demonstrates the risk reward for different therapeutic areas. So really looking at the developmental costs and the potential profit that developers could potentially make for different kinds of medicines. So of course, at the top, we've got cancer treatments and respiratory treatments, which, like other therapeutic areas, cost an awful lot of money to develop, but the potential profit, should you be successful in the developmental process, is potentially sort of blockbuster revenues. However, right at the bottom there, we can see antibiotics, which also cost a lot of money in the developmental process, but are virtually making no revenue whatsoever for developers upon success and market entry. So what is the role of SMEs really in AMR innovation? So in a very timely manner, the World Health Organization has just republished their report on antibacterial agents in clinical and preclinical development. So this is data submitted to the WHO last year in 2023, and this report is incredibly useful, as not only does it tell us about all of the different agents which are in particularly clinical development, but it also gives information as well on the preclinical development pipeline. So I've just taken some of this data here really to illustrate the kind of companies and organizations who have preclinical pipeline antimicrobial agents. And really what we can see is that greater than 95% of the organizations developing preclinical products in antibacterials are micro, small, and medium-sized enterprises, so SMEs, with less than 5% of the organizations being large companies, so such as Big Pharma. Now, I should caveat this with, of course, Big Pharma doesn't necessarily reveal what it's in its preclinical development pipelines for any therapeutic areas, and antibiotics, of course, no different. But this data does at least give us a feel for the significant contribution of SMEs in preclinical AMR innovation. And so what kind of work are SMEs doing in the drug development pipeline for antimicrobials? So I've just got a very simplified view here of the drug development pipeline for an antibiotic. So in the very early stages of development, we have that feasibility work. This could be looking at discovery of hit molecules, it could be looking at the validation of targets, and so on. And then we move into early preclinical development, where we're making those hit molecules potentially more drug-like. And if we can successfully navigate this phase of development, that enables us to select a preclinical candidate for, hopefully more than one, for onward development through those animal toxicology studies, which are also called CTA or IND enabling studies of development. And during this phase, we can also do PK/PD work, formulation studies, and so on and so forth. And then hopefully one of your preclinical candidates can make it through this phase of development, enabling you to select a clinical candidate for which you have confidence in to put it into human trials effectively through phase 1, phase 2, and 3. And then hopefully, eventually, you end up with a product which you can submit to regulators to launch your product in a given territory and get those important new medicines to patients. So early feasibility work is largely done by academic groups. Some SMEs and Big Pharma do have activity in this space as well, which Alisa touched upon. But SMEs largely fit into this middle ground here, working from that kind of early preclinical stage of development all the way through to early clinical trials. And largely SMEs at this point would look to do a deal with a larger organization such as Big Pharma, who have those deeper pockets to fund those pivotal studies, and also the existing infrastructure and marketing teams to commercialize the product as well. So I mentioned at the beginning of the talk about the broken market in AMR, and it's really important that we continue to incentivize developers to make more antibiotics effectively. And this can be fulfilled by a couple of funding mechanisms. So we have push funding, and this is really the funding. It can be grant funding, non-dilutive and in-kind funding to get those hit molecules and candidates through the drug development pipeline to launch. So CARB-X and other organizations are a good example of push funding. And then at the other end of the development pipeline, once we've got onto the market, we have a mechanism called pull funding. And this is effectively the treasure at the end of the rainbow, the carrot on the stick, if you like. So this could be a number of mechanisms, but it would ensure that developers are reimbursed for their development costs upon entry into the market. So a good example of this is the recently launched UK's mechanism subscription style model, where developers would be paid a subscription fee for access to their important new antibiotics. And this would be delinked from volume of sales. So up to £20 million would be paid per year to developers for access to their new medicines, regardless of the volume of sales. Okay. So if we have an idea for a new antibiotic, what do we do? So the first port of call is really to think about the final product. So we need to think about what patients we are going to treat with our new agent, what infections do we need to cover, what organism, what spectrum of activity do we need to have in our final product, and how is it differentiated from competitors or standard of care. And using that information, we put together a document called a target product profile, or TPP. So you may have come across this before in other webinars or indeed at conferences and so on. But this document is effectively a living document that travels with your developmental antibiotic through the lifetime of its developmental process. And it effectively describes what your product should look like once it gets to market. And then alongside that, we develop another document called the Target Candidate Profile, or TCP, and this effectively describes all the different criteria which your antibiotic needs to meet, those different parameters, and a variety of go and no-go decisions for your developmental project. And feeding into this, we also have assay cascades or assay funnels, which form the early stages of our preclinical development process. So here we just have an example cascade that we may use in preclinical development in a hit to lead or lead optimization process. So the assays that feed into our TCP are effectively organized into a cascade or funnel and separated by a variety of go and no-go gates, if you like. And the idea is to triage your compound, so you effectively, hopefully, end up with at least one preclinical candidate for onward development. So the assays tend to start in a quite high throughput and inexpensive manner to run, and they tend to get more complex and lower throughput as we get through the cascade. And we also introduce some in vivo or animal work, which are just identified by the red borders there. So things like PK, efficacy, and sort of non-GLP tox towards the end of our lead optimization process. So what are the challenges for SMEs in AMR R&D? Now I'll caveat this with, this is very much from my experience working in UK biotech, so the challenges faced by organisations in the US, Europe, or different parts of the world may be different. So funding, this is the first challenge. Of course, funding is a key challenge for most areas of science, but particularly for SMEs in AMR R&D. And that's largely because, unlike bigger organizations like Big Pharma, who may have a blockbuster oncology asset which is marketed, and the profits from that can fund other areas of the preclinical portfolio for that company, SMEs in AMR tend not to have that regular revenue stream. And thanks to the broken market in AMR, there's also a lack of early stage VC investment in this space as well. And this leads SMEs to be extremely reliant on grant funding, non-dilutive funding, and in-kind funding. And of course, whilst we're waiting for the decisions of grants that we've applied for, this can create delays for programs where they may be paused, waiting for that next injection of cash to get it to the next value point. And if you're lucky enough to be one of the folks applying for grants or looking for investment in SMEs, you can unfortunately find yourself spending more time trying to raise money to fulfill your programs than on the actual science. And linked to this as well is a lack of exit routes for SMEs in this space. So as I mentioned earlier on in the talk, SMEs don't generally do commercialization themselves, but really look to do deals, perhaps out licensing or acquisitions with larger organizations such as Big Pharma, who have those established marketing teams and infrastructure for commercialization of those products. However, again, thanks to the broken market in AMR, there really is not enough major pharma acquisitions, even of clinical stage programs in this space, although there are some exceptions to this. Then we move on to risk, and this is largely around portfolio size in a small company. So if you have a large portfolio of programs, perhaps they're all diverse in their technology, their modality, or their TPP, this is, of course, lower risk for the company as you cast your net wider in terms of technology. But however, it is more expensive to fund all of these programs. And so we can find that progression of each one of those programs in a large portfolio can be relatively slow. On the other end of the spectrum, SMEs may have just a single asset, or they will have a single technology which has yielded a platform of a few programs. And of course, it is easier to fund a single project or a platform technology, but of course, it is much higher risk for the company, as if that single asset or that single technology is found to have a toxicology flag, for example, this can be the demise of the technology, and unfortunately, sometimes the demise of the company as well. And then finally, capability. So of course, drug discovery and development is a multidisciplinary and highly complex process. And as a small company, it is extremely difficult to have all of that expertise and experience in-house. So it's really important as a small company, particularly for startups, to build that network of expertise through consultants and experienced CROs, so you have all of the bases covered for the various elements of drug development that you need to bring your new product to patients. So now just moving on to a couple of case studies from INFEX. So just briefly to tell you about who we are. So INFEX Therapeutics is a SME based at Alderley Park in the UK, which is just south of Manchester, and we have around 20 employees. We have a portfolio of programs which is very much focused on infection, particularly surrounding pathogens that have pandemic potential. So AMR features incredibly highly in our portfolio of programs, and we also have a couple of antiviral programs targeting coronaviruses with pandemic potential as well. The programs on which we work range quite broadly in the drug development pipeline, all the way from hit finding projects through to phase 2 clinical trials for projects to achieve clinical proof of concept. And in-house, we have experienced teams of microbiologists and synthetic and medicinal chemists. And we've managed to establish ourselves an expert network of consultants and CROs who are largely based at Alderley Park with us, but some are, of course, further afield. And we've been lucky enough to secure a variety of non-dilutive and in-kind funding for our programs through organisations such as the Infection and Innovation Consortium over in Liverpool, through CARBEX, through some of our collaborators, Innovate UK and NIAID based in the US. So case study 1. So this programme was conducted as a collaboration with the UK charity LifeArc and was really born out of a project looking at novel targets in clinically important gram-negative bacteria. So TRIM-D, the enzyme TRIM-D, was identified as an essential and promising target out of this work. And this project was supported by some really excellent structure-guided drug design support as well. So the findings from this project have just been published, and this is in the paper shown here, should you want to go away and look into this in more detail. So extensive effort was put into optimizing enzyme potency for this novel target, alongside looking at antibacterial activity in terms of MICs, as well as cytotoxicity in mammalian cells. And in terms of the medicinal chemistry program, it was incredibly successful in managing to drive down those IC50 values and make some incredibly potent molecules versus the TRIM-D enzyme, virtually down to single-digit nanomolar potency. However, despite exploring numerous chemical series, we were unfortunately not able to translate that potent enzyme inhibition activity into MICs, or antibacterial activity. And this really threw into question the essentiality of the target, or perhaps the druggability of TRIM-D as an antibacterial target. So unfortunately, the decision was taken to terminate this program, but we have, of course, now published these findings for others in the community to read about our observations. And then case study 2. So this is our metallo-beta-lactamase inhibitor program. So gram-negative bacteria producing these resistance determinants, metallo-beta-lactamases, have been identified as a key gap in the antibacterial pipeline by the WHO. And this is because there isn't any existing inhibitor of these enzymes. So based on this key unmet medical need, INFEX Therapeutics set about a development program to develop a metallo-beta-lactamase inhibitor that can be used alongside beta-lactam antibiotics to restore their activity in otherwise resistant bacteria. So we've now published our findings in the paper shown on the top right-hand side here, and in the table below is some MIC data for our lead molecules against a panel of key gram-negative bacteria that produce metallo-beta-lactamases, NDM, VIM, and IMP. And as we can see, the lead molecules 272, 276, 364, and 439 don't have any intrinsic antibacterial activity in their own right. However, when they are used alongside meropenem, which is just shown as MEM there in the table, we can see that these lead agents are able to significantly reduce the MIC of meropenem in these isolates. So based on this promising data, the decision was taken to progress the lead molecule 272 into a mouse thigh infection model to explore the in vivo efficacy of this molecule in combination with meropenem. So as I say, this is a mouse thigh infection model, and in particular, this is the mouse is infected with an E. coli strain, that produces NDM. So I'll just draw your attention to the meropenem group in the middle, where the mice have been administered a dose of meropenem at 50 milligrams per kilogram. And really what we can see is that meropenem alone at this dose is virtually static. So it does not lead to any significant reduction in the bacterial load of the thigh tissue compared to the pre-treatment group. However, when we combine meropenem with the lead molecule 272 in this case, we're able to restore the activity of meropenem and significantly reduce the bacterial burden within the thigh. So based on this promising data, the decision was taken to undertake some additional optimization work, really to improve parameters such as solubility. And this has now successfully led to the selection of the preclinical candidate, which we've called METEX. And thankfully, METEX has now also managed to successfully navigate those CTA and IND enabling phase studies, so those toxicology studies in animal models. And METEX, in combination with meropenem, is now ready for those first in human studies in a phase 1 clinical trial. So just some final thoughts before I finish. So SMEs really do the majority of the heavy lifting, particularly in preclinical innovation in AMR R&D, but they do face numerous challenges along the way in this endeavor. And this is largely around reliance on non-dilutive and in-kind funding, a lack of exit routes, even when they've managed to get their products through to clinical development phases, It can be challenging to manage risk as an SME, largely around portfolio size, and it's really important to build that robust network of experts to make sure you've got all of those bases covered for every element of the drug discovery process. However, I would say from experience, if you have a great team of scientists and have some really innovative programs, and you're prepared to be very flexible in your way of thinking and can be eternally optimistic, then these challenges can really be overcome. And with that, that just leaves me to thank you all for listening, and of course happy to take your questions, and of course thank you to everyone who was involved in these projects as well.
Thank you, Vicky. We will now start the Q&A session. As a quick reminder for the audience, you can submit your questions as shown on the slide, and we'll do our best to respond to as many questions as possible during the allocated time. I'll start off with something that is probably considered to be easy. It's about the early characterization and MIC testing, and the question is that how important it is to consider alternatives to just the normal protocol used, outlined usually in CLSI or UCAST guidelines, and how do you deal with the fact if it doesn't actually if it doesn't work with those standard protocols? And I think the question is for you both. Whoever wants to go first.
I'm happy to start. I think so. So your question is really around if you have molecules that may not really work in your standard MIC assay that CLSI is method wise, and so how. That's a great question. I mean, I think that's something difficult to face because we tend to think of the microbroth dilution, macrobroth dilution as the standard assays, and perhaps your drug may require specific, additions, iron components to the media. I think that it's still acceptable. So if you have an understanding of what is really required to get to the bottom of your MIC testing methodology, it's okay to develop some sort of slightly different method. That may become more complex when we think about having clinical labs being able to test your drug and perform susceptibility panels. That can get more complex when we also think about susceptibility testing devices, which can sort of build on the complexity if that's required. But I think that understanding at least whether or not you need to add special components, incubate differently, different temperatures, is an important thing to work out early in your drug discovery process, so you're able to continue to test that molecule accordingly. And Victoria, I don't know if you want to add on to that. That's
Yeah, sure. So I think it's probably worth saying at this point, particularly here at INFEX, we have a variety of programs, many of which are not direct acting antibacterial. So we've had virulence inhibitors, for example, where MICs are just not applicable at all. So in those cases, we've had to develop assays kind of from the ground up to best evaluate those molecules for development. So it's absolutely essential for some programs to develop something else where an MIC isn't useful. Having said that, talking about direct acting antibacterials, of course, we prefer to stay with the guidelines, the gold standard CLSI where we can. And I think it's really important to understand and have that evidence, particularly in vivo, that says a change from the gold standard is required. So, for example, Capidericol is a good example of that, where a whole new diversion in the CLSI guidelines had to be created because the MICs, as they were done, are not reflected of the potential of that molecule in an in vivo setting. So I think in some cases it is necessary, but absolutely there has to be strong evidence for deviation from those sort of standard protocols, I'd say.
Mm-hmm. Yeah, and then I think in the end, when we think about sort of the applicability of testing your drug and being able to help guide therapy, I mean, that then builds on complexities. And so, you know, I don't think there's one answer. There's not one answer that would fit everything. And so that's also something important and to consider as well.
All right, thanks a lot to both. So I think I'll just use that as a segue to the funding question that that I have on it. Is basically you're dealing, or you have dealt with, a lot of different funders, funding agencies, charities, and so on at almost all stages of your programs. And so in a way, it's also what is important to show to them, and which sort of, like, let's say, the examples of how to deal with them at the different stages of a program. Maybe Vicky, you can start, and Alisa, you can go second.
Yeah, I mean, I think for most funders, it's reasons to believe, isn't it? So you have to provide the necessary information that gives them reasons to believe and effectively invest in your program. But I'd say certainly as you get further into the process, as you get closer to clinic, if you're applying for grant funding to get through clinical trials, being very, very clear on your clinical development plan and how you're going to be successful through that process and navigate regulators is really important. And particularly in early stages of development, if you're working on a modality that's been found to have liabilities for toxicology, how you're going to build in the findings of others to give confidence that you're de-risking your approach from others who've gone before. So those are probably some of my experiences. I don't know if Alisa wants to build on that.
Yeah, so I think also, you know, different groups may or may not want to be a little more hands-on, and so I think that's something to consider with an SME. And so I have a more recent experience. I've worked with previous funders in the past at occasion, but that's been almost a decade ago now. But I can say, you know, some agencies really like to have continuous updates on progress. So you might think about monthly written progress reports, monthly project meetings, interactions with typically these groups, and I'm thinking of BARD in particular, they have you know, wonderful teams that they set up that have a core group of SMEs, and in this context, subject matter experts and not small medium enterprise, but they have a wonderful set of SMEs that also work with us. You're really thinking about a partnership. There's often face-to-face meetings, and there's a lot of back and forth. So it's great. It feels like an expanded team, and you've got these experts to interact with that also help that knowledge base of your program. But that's definitely something to consider, because there's a lot of what you will consider administrative work when you enter into these contracts and agreements for you know, ensuring that you're making that forward progress, as you mentioned, Vicky, as well.
All right, thanks. Next couple of questions are more like in the later stages, or considering later stage aspects. One particular question is, if one considers the example of plasmicin, how do we de-risk a program early to make sure that it will be profitable?
That's a very important and difficult question to answer, and I think Victoria captured it nicely on one of her first slides, talking about the AMR market being broken. And so I think that, you know, we both described understanding that target candidate profile, target product profile, and understanding that throughout the entire course of development. And I think that, you know, ensuring that the indications that you're seeking, that there's actually a place for your drug there, and that, you know, there's going to be utility. And I think so that's one aspect of maybe earlier in development, where you can ensure that sort of you're going to not only meet an unmet need, but also be a valuable resource at that later stage post-approval. And then I think, you know, thinking about pull incentives as well. And Vicky, I don't know if you've got some additional context to add here.
Yeah, I mean, I think talking about direct antibacterials, antibiotics, I think pull incentives are going to be so important to make the area profitable, if that's the right word, but really to incentivize big organizations and more sort of SMEs to get involved in the space as well, to ensure we have that continuous supply of new agents coming into the clinic. I'm not sure we can fix the reliance on volume of sales to guarantee reimbursement for companies. I think pull incentives will be really, really important in this space. So I mentioned the UK mechanism, which is now being rolled out by NICE, but of course there's the Pasteur Act in the US as well, which will be so important to kind of reinvigorate the space and kind of pull more big organizations back in and sort of reinvigorate the kind of investment space as well, I think. So, yeah.
All right, the next one also on late development, and maybe I'm not sure how we can tackle it, but I'll just put it out there. So if you want to develop an already approved antibiotic for new indication, so what type of requirements do you have, or do you have to fulfill? I mean, this is more a regulatory question, but maybe Alisa, you can give a go, and then Vicky, you can comment if you want.
Yeah. Give a go. So first, I think it depends on whether or not that antibiotic is patent protected, of course. So let's say it's been on the market for decades and it's a generic and you're looking for a new indication. So, understanding the available safety database is going to be critical at that point. I think that so to your point, this is probably a regulatory question, but I can come up with some ideas. You'd obviously still need to, in my opinion, demonstrate efficacy in the indication that you're seeking. Is there going to be a different formulation? That's also going to be an important thing to consider as well. Are you going to be looking for this drug to be utilized alone? Are you thinking of combination agent? I mean, there's a number of questions that are sort of wide open and out there. But it is perhaps an advantage if you do have, if that drug has a long-standing history of safety and known liabilities, known things that you need to monitor, that there is a place where perhaps you may be able to get away with a slightly smaller package, basing your package on the foundation of what's already known. So Vicky, I don't know if you've got any additional sentiment in that.
No, probably not. I think that covers it all. It's just having an awareness of what the additional gaps you might have to fill in terms of development to get into that new indication space. But no, I think that's pretty well covered, Alisa.
Thanks.
Okay. Next one is, I think everybody of us can relate to that. If the MIC is on the higher side, e.g. MIC 16 or MIC 19 around 16, but performs well in vivo, what's path forward?
Good question. I suppose it depends on the modality, doesn't it? When we're talking about peptides, maybe an MIC of 16 is pretty decent. If we're talking about small molecules, less so in terms of actual number of molecules available to the bacteria. For me, in vivo data would trump the MICs, but it's trying to build an understanding of what your in vitro, in vivo correlation is, what's your driver for determining efficacy, and then building that into your preclinical development package. So yeah, I'd say that the in vivo data definitely trumps the in vitro data, but going forward, you will be reliant on that in vitro data for PK/PD work, and eventually when you get into clinic and so on. So you do need an understanding of why there is potentially a disconnect between your MICs and your in vivo situation. And perhaps coming back to the first question, if any modifications to that MAC protocol might be more correlative with your in vivo data.
Mm-hmm. Yeah, I think that's answered well. I mean, I don't have too much to add, just an understanding of what that PK/PD driver is. Perhaps that can be covered easily. We think about, if you take a look at the CLSI breakpoint list, there are some drugs with high breakpoints, aminoglycosides, for instance, and I won't comment on whether or not those are still accurate. There's a question there, but they are quite high, and so that could be an example where perhaps an MIC of 16 is not of concern. But I do agree with everything you said, Becky. I think that's important.
And looking at PK as well, perhaps 16 micrograms per mil is easily achievable, but by your particular drug that you're you're developing. So perhaps it's not a problem, but yeah, you need that understanding of it to move forwards, I'd say.
Yeah.
All right. Next question is kind of like one of those. What is it a good target? What you're working on? And the question is specifically on lipopolysaccharide biosynthesis targets. So LPXA-C, LPXD. Considering that there's basically like a Acinetobacter that can do without, at least in vitro. Or that there's like resistant ones that seem to lose at least parts of the LPS and still seem to do quite okay. So your comments or your thoughts on whether LPXC or these basically LPX enzymes are good targets?
I can start that since I brought it up, I think. Yeah, that's a difficult question. So I think, if we look at the history of the LPXC pathway and the interest in it, I mean, I think there's still a lot of interest. There's still companies out there that I know of that are pursuing LPXC inhibitors. And so I want to say yes. I mean, I think that there's still potential there. There's, it is essential in most gram negatives, yes, but you're right about the Acinetobacter issue, and I think that there's such an incredible foundation now. I refer to the Alice Irwin review, which ended at 2016, right? So we're almost a decade later. I think that there's additional reviews out there on this target. So I would say that optimistically, like, I do think it's a good target to continue to pursue, and I think that that's probably why there's still a lot of interest in that target. Now, there are liabilities that we know about some of the molecules that have been tested in the clinic, but we're continuing to build that knowledge base and just continuing to build that foundation with publications. So hopefully it can be a problem that's solved. Vicky, I don't know if you've got any additional.
Yeah, I can't really comment on LPXC specifically, as it's not something that we've pursued. But I would say, kind of based on kind of the stuff you presented, Alisa, as well as some of the case studies that I presented, it is difficult working on novel targets with novel chemical scaffolds, novel modalities. It's high risk. There isn't that precedent already in the clinic or even in clinical trials necessarily. So your new chemical series is at risk. You don't know the tox profile of that. You don't know the profile of your novel target, but it's high risk but potentially high reward as well. So it is worth it, but you will see more attrition probably in those projects versus those which are well established, like aminoglycoside derivatives, for example, and beta-lactam, new generations of them. So definitely a more high risk approach to drug development, but potentially higher rewards as well.
All right, thanks. Next question, and we're going now really into covering lots of different targets, even those that you didn't actually sort of cover. So it's about virulence factors, or I would also maybe call them potentiators, because it basically says in the question, synergistic mechanism with class of antibiotics, but obviously don't actually have antibacterial activity on their own. And the question is specifically about, okay, do you need to show with each individual antibiotic in vivo combination, or are there shortcuts that you could do to go to the clinic faster with something like that?
Big question. Yeah, I can take that one on. So I suppose if we think about potentiators, we can sort of put them into different buckets. So we have the resistance breakers, which are pretty well established through beta-lactamase inhibitors. So I talked about our metallo-beta-lactamase inhibitor program, and beta-lactamase inhibitors are really well used clinically. There's a variety of combinations of beta-lactamase inhibitors with beta-lactam antibiotics. Now, the current approach to development is through a fixed dose combination. So you can't just take that beta-lactamase inhibitor and put it with any old beta-lactam. And so true is for potentiators where you'd want to use it as a standalone agent for use with multiple antibiotics. So far, it's sort of uncharted territory that no one has gone down yet. And the same, of course, is for virulence inhibitors. So I mentioned that we have worked on some virulence inhibitor programs. So there are sort of numerous challenges along the way. So I mentioned about working on novel antibacterial targets with novel chemical series. You know, when we're working on these novel modalities, which could potentially be a game changer for AMR, and we're sort of finding our way as we go through the development process, and a lot of engagement with regulators has to occur to facilitate that process. I don't know if you've got anything to add, Alisa.
No, I think that's great. Thank you.
Good. Next one, I think it's a tough one. So it's about sepsis or septic shock, and basically the role that the drug developers could play in bringing new modalities, or also studying even the known ones. And then the example that is given here is carbapenems to patients, and also especially also in LMIC countries. So did you ever have any thoughts on whether going into indications that where the patients with septic shock or with sepsis are involved, and do you consider that to be a feasible strategy as a drug developer? I think the unmet medical need is huge, we all agree, but from a drug development point of view.
I can only offer a side note. You're right, this is a difficult question. Achaogen did have a program previously partnering with the Bill and Melinda Gates Foundation, and potentially there were other funders, I can't recall, but there was a program looking at extended spectrum aminoglycosides. So not plasmized, and potentially modifying new aminoglycosides, looking particularly at neonatal sepsis, because when we think about that, I think gentamicin might be often sort of there. And so, unfortunately, that program obviously was terminated at the bankruptcy as well. So I'm not sure how far along things really got, but I do think that that's absolutely an important question for drug discovery folks. And Victoria, I don't know if you have anything else to add.
No, not really anything to add, I'm afraid, on that one.
Yeah, I think I would just direct whoever asked that question. You could probably Google and find a little bit more about the Achaogen program to see, what was done there. I'm assuming there's some information online, at least with press releases.
All right. The next question puts you on the spot, Alisa. It's actually a very specific question. What do you prefer, a broader market with more competitors, or a smaller market that you own?
It's a specific question to me. A broader market with more competitors, or a specific market.
Small market or a specific market that you own?
I'm going to answer this the way that my husband answers questions like this, which is I don't have a favorite. I think that there's probably an avenue for either, and I think it really depends on the landscape. Can you really fulfill some unmet need? Is there a broad market, yet issues with resistance with those drugs that are considered standard of care? Has a standard of care not actually been updated in years? And so is there a place where your drug can fit in? And then a smaller market we can think about, that you own, but then of course that also may compromise your commercial success. Is there an issue there with being able to maintain and sustain your market in that event? So that's sort of my easy out there.
Even though it was directed to you, I think Vicky, you can also maybe.
Yeah, can I say I don't have a favorite either, just a market that has space for antibiotics in it that are reimbursed properly would be my favorite.
Yeah.
All right, good. So next question is on kind of like academia versus SME. So considering that a lot of foundational work is done in academia, so what makes SME better suited to advance discovery programs into early development than academia?
Another good question. I don't know if they're better suited per se. It's just different sets of skills, isn't it? You know, and your goals, whereas an SME has sort of corporate goals to commercialize agents. And, you know, for example, at INFEX, we put a lot of effort into sort of building that network of experts. We're very acutely aware of how multidisciplinary drug development is. So we have those resources available to bring those ideas, those feasibility kind of findings through to something that's commercializable, hopefully. So I'd just say it's not that one is better than the other. It's just that the sort of your agenda and your goals are very, very different in a commercial enterprise than they are in an academic setting, I'd say.
Yeah, and I can add to that and say that, you know, I think we also shouldn't think of these entities as sort of separate. You know, Paratek, and also when I was at Acadian, we collaborated really nicely with a number of academic institutions. In some cases, you may not have the expertise at all to run certain assays, or not even have a lab yourself. And then you can find and interact with these academics to help build your program out and you know, conduct some really critical research. And so that's how I've had interactions with academics in the past as well.
Yeah, I think that's a really important point. You know, we have some really great collaborations with academic groups, and that's so true. Whilst we don't have the bandwidth to do some of the really exploratory science, as we've got to focus on moving our programs forward, you know, some of our collaborators have been instrumental in doing those kind of studies for us, which has really added value to our projects as well.
All right. I think the next one, it was one of the examples that you brought up, Vicky, that triggered this question was comment on the rational design approach, identify inhibitors, and then trying to optimize for antibacterial strategy. So if you could just kind of like elaborate a little bit on probably on how successful that is generally, or what your thoughts are on it.
Yeah, I mean, I think, you know, structure-guided drug design is a really important component of a drug development program. But, you know, personally, it's been really important and valuable to us to try and optimize different parameters in parallel, rather than just trying to focus on your enzyme activity and then going down antibacterial activity. It's important to try and consider both in parallel. So we don't just focus on kind of optimizing the enzyme potency, particularly with gram-negative bacteria. We're trying to optimize features of those small molecules that promote things like entry into the bacteria and prevent efflux and things like that. So for me, it's important to consider both simultaneously. And then as you get further into your drug development program, or perhaps earlier on in it, you may need to optimize other components as well, like solubility or things that improve your stability, clearance, and so on and so forth. So there's multiple things that we have to think about optimizing in a medicinal chemistry program. I'm going to stop here because I'm not a chemist and I am a biologist, so I will, yeah, I'll stop there. I don't know if you've got anything to add, Alisa, on top of that.
I think you covered that very nicely, yeah.
All right. Next one's on timelines. So initiation of programs to kind of like getting to the milestones or the go no-go decisions. Maybe Alisa, you can start off. So is there anything that you kind of like, you give yourself some time, or what's your experience and the average time?
Yeah, I think, okay, that's a pretty broad question, so I'll see if I can hone it in. Trying to think of some examples of my previous, the previous programs I've worked on. You know, I think the go no-go decisions are obviously really critical. So let's say you're at a point where you've identified your lead candidate molecule, and now you're going to be putting that through the series of, you know, gating studies. You know, having specific focus gating studies. So if you're working with novel molecule, a novel target, that might be slightly different than if you're working with a drug that has a well-known class, that because you have really specific questions that you might be able to ask versus something where it's a little bit of an unknown. So if you are partnered with a funder, there might be timelines placed on you for conducting these gating studies and making these go no-go decisions. I think that it could happen really quickly. We could think about completion of studies in a couple of years, and when we think about in vitro studies are typically quick turnaround time, but when we're thinking about studies, in vivo studies in animals, depending on even short tox studies, short is a relative term, that might take a few years. And so I think that really ensuring that you make considerate decisions at each stage when the data is available is really of utmost importance, and then following that timeline potentially based on what's communicated to you by your external funders. There may be pressure from senior management about moving things forward, and so that's also important to consider as well. It's kind of a vague answer, but I think it really depends on the project that you're working on and the stage that you're at. But as I mentioned, I think Vicky, you also mentioned this, when we think about the totality of a development program, we're thinking about at least over a decade, typically, depending on what stage you're in.
Yeah. I'd say that's probably best case scenario, isn't it?
If it's optimal scenario.
Yeah, I mean, I think thinking about development of COVID vaccines and stuff, when the medical need was astronomical, but resources and funding were virtually unlimited, I think that gave us a view of how quickly drug development can go from an idea through trials into patients. So coming back to one of my slides, of course, in an SME, a lot of your timelines are highly dictated by the funding available for your given project. So yeah, the answer is a bit, how long is a piece of string? It very much depends on your funding situation, the type of project, what kind of studies you need to do. But certainly that 10-year is an ambitious goal from idea to patients.
Yeah, maybe just to follow up on that, you both mentioned basically the importance of the go no-go decision. So kind of like any advice that you can give on how to set them up properly so that they're useful and that the teams can really work with it.
Yeah, that's a pretty broad question too, and maybe I'll just give examples of some. So I think, for example, I talked about programs that had resistance liability, and so to me, that's as a microbiologist, that's a really critical piece of information that would come out of my group is understanding a resistance liability, and I think ensuring that you're performing the specific experiments that can help you observe a resistance liability, and sort of give examples of what may happen in the clinic. So there's a couple of easy ways to do this in vitro, and one of those signals came up quickly in the biotin carboxylase program that subsequently led to termination, which is you have this huge population of bacteria that then you probe to see, are there any mutations that actually are just pre-existing in the population. So that's a huge concern. You don't want any of that to be a liability. Of course, there would always be actually mutations, but ensuring that you're observing what those mutations are and whether or not you have a huge shift in your MIC and what that mutation is resulting. So that's one example where I think streamline those early on. When you have a lead candidate, really understand the potential for resistance liability. That's going to inform in vivo efficacy studies as well as what you might want to achieve in the clinic. And then I think, of course, tox is an important question as well. When you get there, you can do cytotox assays, but then you're thinking about what those potential tox signals may be, especially for an unknown molecule. Now, as we saw in the LPXC inhibitor program, the tox that was observed in humans didn't really track with what we saw in animals. We had to go backwards and sort of create an assay, and so that's that's something that just came up as an unknown at that stage. So Vicky, I don't know if you have any other insight into this as well.
No, I think that's pretty well covered. I think yeah, quite a broad question. I'd probably just come back to one of my slides early on, just about being very clear at the start of a program what those parameters look like, what those go, no-go decisions are, and be quite firm with them and be quite ruthless. Just, you know, like some of the programs we've talked about, we don't want to get through to a stage of development where it's then extremely costly and devastating sometimes for companies that they end up terminating a program at a late stage. If it needs to be terminated, it needs to be terminated early on. So it's doing those, like you said, Alisa, those killer experiments that are sort of critical path for you to make those decisions early on in the drug development process. So that's all I'd add really on that question.
Okay. The next question is on, let me just find it, sorry. On basically the use of bioinformatics and next-gen technologies, AI, obviously, AI and machine learning, and what role they play in your organizations these days?
Well, I can start by saying that I'm pretty much a dinosaur when it comes to AI, so I probably can't comment on that front. But we do use, you know, a variety of like next-gen sequencing, particularly with a view to understanding resistance when we're doing resistance work in microbiology for our development programs. So it's basically integral to our drug development programs, particularly on that front, and looking, you know, at epidemiology and things like that. But yeah, I'm afraid I can't comment on AI really.
Same for me with regards to AI. And I think, you know, we have our programs are on a molecule that's now approved. So we're not necessarily in that really early discovery phase needing AI. I think it's an exciting, you know, opportunity for those in research and discovery to use those advanced next gen and AI tools. But I don't have any experience on that. And I think back when I was at Occasion, it was pretty early days. So crystal structures and PyMol and all those programs were used at that time for structure activity relationship building.
So maybe let's just kind of like poke onto that. Both of your lead programs are either in clinical development, going for proof of concept studies now, or they're like really late stage. So if it's not AI in drug discovery, so do you use it for patient selections, for indication selections, and these type of activities, or are you aware of those things happening in your organization?
I certainly am not aware of AI being used in patient selection, at least for INFEX Therapeutics point of view. However, you know, there's been a variety of papers published now, kind of early stage papers trying to find new molecules on various antibacterial targets. So I think AI is important in kind of the discovery end of it seems to be. However, I think from speaking to experts in this area of which I am not one, there's still a lot of development of AI to be done to bring in all those different disciplines of drug discovery. It's not just about antibacterial activity and enzyme potency, as we discussed before. There's so many other attributes of a molecule that have to be optimized for it to be anywhere near being a drug that can be given to a person. So trying to bring in all those different elements of drug discovery and apply that via artificial intelligence has got some way to go, I think.
And my answer is the same as Vicky's to your initial question. So I'm not aware of that being used at the sort of patient level.
All right. Maybe next one's going to be almost our last question is public-private partnerships. Kind of like, have you been kind of like part of those? And if so, what experiences are those, or have you with public-private partnerships?
Well, so Paratek was recently acquired by Gurnett Point Capital and Nova Holdings last year. So we were primarily public, and now we're private. So that's actually, I think, exciting way for Paratek to continue its business. He mentioned the broken AMR market, and Granite Point Nova Holdings is really interested in driving success and innovation and commercial opportunities for SMEs generally. And so I think that is a rewarding relationship that we've just embarked on, not just a year ago. I don't know, Vicky, if you've got any additional flavor.
I can just give kind of one example, I suppose. So INFEX is a sort of founding member of the Infection Innovation Consortium, which I mentioned in my slides over in Liverpool. So this is a fantastic sort of consortium collaboration that's been set up in the northwest of England and includes numerous academic groups at the Liverpool universities. It includes a variety of commercial organisations, including us, and also some of the clinical team over at Liverpool Hospital as well. So this has been really instrumental for us in kind of finding further opportunities for our portfolio in terms of clinical development of some of our assets. So for us personally, it's been really important to have that sort of public-private partnership for many of our programs.
All right. So I think that concludes our Q&A session. And I would like to thank everybody for their questions and comments. And once more, and most importantly, thank you to both of our speakers, Alisa and Vicky, and hand it back now to you, Victor, for the closing remarks.
Thank you very much, Daniel, for moderating today. And thank you, Vicky and Alisa, for your presentations.
Thank
you.
You
can already register for our next revival webinars. On 23rd July, we will have a webinar on the value of susceptibility surveillance data, which will also feature information on the Vivli AMR surveillance data challenge. There will also be a webinar on 22nd August on developing non-traditional antimicrobials. Please also make sure to keep an eye out for our newsletters and also follow GARDP on X or Twitter and LinkedIn to hear the latest updates from Revive. So that will be all from us. Thanks for joining today and for contributing to this discussion. I hope you found it interesting and useful and that you'll join us again in the future. Please do encourage your colleagues to join as well. And also please fill in the survey afterwards to inform us on how to continually provide these webinars and improve them for the future. Thanks again and goodbye.
Bye.
Bye-bye.